Yes. Good morning. So, at few minutes remaining is Jain. So, today we discuss the that is male gametophyte and female gametophyte. So, all of you, Jain, please, is audible clear? Anybody?
Yes, sir.
Okay, thank you. So, few minutes we have to wait. Remaining people to join. Right. So, today class we discuss the male gametophyte and female gametophyte development. Structure of that male gametophyte and microsporogenesis, and another female gametophyte and megasporogenesis. Is there anybody? What is the male gametophyte? Is there anybody? What is male gametophyte? Is anybody tell? Yes.
It is
So
developed from
means.
pollen grains.
Oh, sorry.
Developed from pollen grain found
Yeah.
on the
Yes. 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 The male gametophyte is nothing but pollen grain only. So means the male gametes that also known as the sperms. We know that one. That is the you can see the ninth unit. Okay, ninth unit you can see the structure and development of the male gametophyte. Okay, so that the male gametophyte also we called as the pollen grains. So pollen grains are formed within the microsporangium, nothing but anther. Okay, so when we take the here cross section of these microspore are nothing but anther or microsporangium, so which is consist of an oval mass of meristematic cell which is surrounded by the epidermis. So here four lobed are formed in the archisporia and get the differentiated into hypodermally at the four corner of the anther. So see here that is uh, the anther structure. See your that is the ninth unit. So page number 153. That is the structure of anther is there. four lobes are present, right? Four lobes are present. That is the uh, in the archisporal cells undergo to parenchymal division, forming the primary partial layer and the primary sporogenous tissues. Now we discuss the structure of anther. Transfer structure means TS. Transfer section. When we take these fertile archisporium tissues, which is surrounded by the sterile tissues there. So latter is many layered, and each layer has a distinct role to play. So we can see when we take the here transfer section of anther, the anther wall development, the primary partial layer undergoes to periclinal division and anticlinal division, forming the anther wall. On the basis of the formation of middle layer, Davis is classified the development of anther wall into four types, that is basic, dicotyledons, monocotyledons, and reduced types of the development of anther walls. So what are the basic type of anther wall? Means primary partial parietal layer undergoes to periclinal division, forming the outer and inner secondary parietal layers are there. So both layers undergo to periclinal divisions, and one towards the epidermis differentiation to endothelium, followed by the middle layer, and other secondary secondary parietal layers produce the another middle layer and tapetum. Another type is the dicotyledonous type. The primary parietal layer undergoes to periclinal division, forming the two secondary parietal layers. Of these, outer parietal layer divide periclinally, gives rise to the endothelium and middle layer, while inner layer function directly as the tapetum. Monocotyledonous type of means epidermis, that is the primary parietal layer undergoes through the periclinal division, forming the two secondary parietal layers. So from this outer layer directly function as the endothelium, while the inner one undergoes to the periclinal division, forming the middle layer and tapetum. So another reduced type of epidermis is there. Here also, that is the primary parietal layer undergoes to the periclinal division, which is formed the two secondary layers are there. Outer one directly function as endothelium, inner one to formed as the tapetum. So when we take the anther wall structure, means here there are the four categories, four parts are there when we take the anther transfer section here, that is the epidermis, endothelium, middle layer, and tapetum. Okay, four parts are there, that is the epidermis, endothelium, middle layers, and tapetum. These four walls are there. So the epidermis is a single layer and is the external covering of the sporangium, microsporangium. Epidermis is the outermost layer, single layer, which is the protecting layer, and which one is the cover to the microsporangium. The epidermis at first keep pace with the development internal tissue by the anticlinal divisions, but the due to the pressure extended by the developing cell inside this progenious tissue became the rapidly radially flattened and stretched both tangentially. So it is the protection function. Epidermis, what is the main function of epidermis? Which is protect the internal organ. So below the epidermis, endothelium layers are there. So the layers of cells lying immediately beneath the epidermis. Below the epidermis or beneath the epidermis, we call as endothelium. The cells which is very enlarged and develop fibrous thickenings are present their inner tangential wall at maturity time. But here, endothelium mainly involved in the dehiscence of these anther wall or breaking the anther wall. Means these endothelium cells only fibrous thickenings are there, large cell than compared to remaining all cells. So these bands extend rapidly toward the upside, terminating near to the outer tangential wall. Means fibrous bonds are there. So these are extended to the outer side. So thickenings are usually absent in the region of dehiscence. So these endothelial thickenings containing mostly alpha cellulose fibrous thickenings are absent in the clistogamous flowers. Clistogamous flowers means, is anybody? Clistogamous means phanerograms.
Flower forming or not flower forming?
So that is the not blooming flowers. Clistogamous means that cannot bloom. That one we call as a clistogamous. Clisto. Okay. So here that is the means there is no cross pollination in that clistogamous flowers. Means example Comilena bengalensis, that is the non-blooming plants and always in the self-pollination only. Okay. So here the presence of fibrous bonds, the differentiate expanse of the outer and inner tangential wall, and these due to the hygroscopic nature of these endothelial cell, which is helps in the dehiscence of anthers at maturity time. So these fibrous thickenings bonds also they have the hygroscopic nature. Hygroscopic nature is there, so that's why they are the involved in the dehiscence of the anther. Means they are observe the moisture their surrounding and that cells will be swollen and immediately split, and so pollen grains are spread into the their environment and which are involved in the cross pollination. We call as the middle layers. Epidermis, endothelium, middle layer, and tapetum. So middle layers. Here, the initial number of middle layers are formed in many anther depend on the type of wall formation. So here, in some families, the middle layer undergoes to the further periclinal forming too many middle layers are there. Some are 2 to 4 middle layers are formed. So here, as the cells of middle layers lack the ability to divide anticlinally, the tissue cannot adjust itself to the presence of extend by the multiplying the and that is expanding sporogenous cells within the sporangium. So the cells of the middle layers ultimately became flattened and crushed at the time of meiotic divisions in the pollen mother cells. In many species, the cells of these middle layers are the storage center for such as starch and oil bodies also there. So the innermost layer we call as the tapetum. This one is a very important nourishment tissues. So the tapetum is the innermost layer of the anther wall and surrounded the sporogenous tissues. The tapetum is composed of a single layer of the cells which are full the dense cytoplasm and prominent nucleus is present. The tapetum is an outside it is derived from the partial layer with the towards the connecting side of the development from connecting tissues. So with the result is the surrounds the sporogenous cells compactly. All the cells are another tapetum are similar in the size and morphology in most of the plants. But in some families like Lamiaceae, the cells are another tapetum or the dimorphic condition also. So in angiosperm, the anther tapetum, there are the two types are that glandular and another one is the amoeboid tapetum. Glandular tapetum and another one is the amoeboid tapetum also present. So means these are the secret the in the pollen grains provide the nutritions. Okay, so these are the so role of the anther tapetum. Main what is the main role of the anther tapetum? So here in the tapetum is of the considerable physiological significance because all the nutritional material entering to the sporogenous cells. So during the meiosis, the tapetal cells simply transport nutrient to the myocytes. So another application here that is the release of the callose. So in this type of enzyme is diplomeres, the callose walls of the tetrads and microspore are released. So another one is the male sterility also. So formation of the non-viable pollen grains in the male sterility lies is associated with the non-functional or faulty development of tapetum. So in the tapetum plays a very important role in the formation of the pollen wall. Probiosco means pro-eubiotic bodies are there. So which is are produced in the secretory tapetal cells that is migrate to the cell surface with they are get coated with the sporopollenin and related to released to the anther locule. So sporopollenin that is the very hard structure. So which is resistant to the some environment factors means tolerant to the heat, salt, that is very resistant to the these environmental factor, salinity and salt, acidity, temperature also. So pollen kit substances and trypsin are transferred from one tapetal cell to the pollen grains. So the pollen wall that contain the proteins derived from male gametophytes as well as the tapetal cells. These proteins are responsible for the incompatibility. The tapetum synthesize the certain substances which are released into the locule during the maturation of the pollen grains. So these proteins enter into the exon of the pollen grains and are stored in the region of bacula. So coming to the microsporogenesis. So see here that is the male gametophyte development in the angiosperm are nothing but microsporogenesis. How the microsporogenesis are formed? So primary, the sporogenous cells give rise to the microspore mother cells. In some plants, the sporogenous cells function directly the pollen mother cells. Example, Boerhaavia. In some other cells undergoes the transfer division to resulting the form the pollen mother cells. So there are the to form the sporogenous tissues means spore mother cells undergoes to the meiosis to form the microspores. Nothing but pollen grains. So if the enter into the meiosis, the pollen mother cells, meristematic cells are there. The pollen mother cell is another locular interconnected by the plasmodesmata. So these plasmodesmatal connections also known as the stage between the tapetal cells and pollen mother cells. When the pollen mother cells enter into the meiotic, that is the plasmodesmatal connection between the pollen mother cells and tapetal cells, the last to the deposition of the callus. So here the pollen mother cells undergoes the meiosis division to form the individual isolated cells are there. The callus walls are formed between the diodes, between the microspore of the tetrads. So here that is a cytokinesis means cytoplasm division. There are the successive division and simultaneous divisions are there. Two categories are there. Successive division means the pollen mother cell undergoes to the meiosis. One which is followed the wall formations are there. Meiosis one that meiospores are formed wall formation. So the cell plate is formed in the center and extend laterally. So the two diode is under the meiosis 2 and wall is formed at the initial resulting the tetrads are formed. These successive divisions is common in the monocots. So coming to the simultaneous division, the first meiotic division is not accompanied by the wall formation. So first meiosis, the wall formation between these mm, myocytes are not formed. So after meiosis 2 only the callus wall formation. Okay. So that is the simultaneous cytokine is common in the dicots. See, male gametophyte germination. We can see in the that is the figure number 9.3 here simultaneous division and tetrads are formed. So just few minutes. So, so that is the development of male gametophyte. Here, so the newly formed microspore has a dense cytoplasm with the central situated one nucleus is present. So microspore after is released from the tetrad, that is a single pollen grain, so rapidly increased in their value. So vacillation occurs the pollen pushing to the nucleus toward the one side. So vacuole is formed and that nucleus will be going to one side of the inside of the pollen grain. So for here the formation of vegetative cell and generative cells are there. Here after releasing the pollen grain from the anther, they are the nucleus undergoes to the mitotic division to form the new two types of cells are there, that is the vegetative cells and another one generative cells. So vegetative cell to give the nutrition to the generative cell. So after generative cell again divide into two, they are fused with the that is central nuclei and egg. So that one is formed as a zygote. So we have to discuss here. Uh, so that is the interval between the end of the meiosis. The first mitotic in the microspore is very short. So in the mitotic division in the pollen grain is locule is synchronized. Those plants in which is the microspore remain the tetrads. All the spore in tetrad are in the same stage of division, but not all the tetrads have the same locule. A complete synchrony may be expected where the microspore are united within the pollinia or Masle, nothing but in Astipidaceae family or Mimosaceae family, these are the all pollen grains are aggregated to formed as a pollinia. So the nucleus pollen grain move toward the side where the undergoes to the division. There is there are two types of cells, generative cells, but may be cut off toward the inner side. Okay. So here the curved wall between the uh, intine and plasma lemma of the generative cell, the mitotic division are always unequal, resulting the large vegetative cell and small generative cells are formed. See here. So this is the large generative cell and small means large vegetative cell and small generative cells are formed. So this is the vacuole, large vacuole is there. Okay, see here, this is diagram. So the generative cell gets the separated from the wall of the pollen grain. They are coming to lies the cytoplasm of the vegetative cells. So vegetative cell after the pollen mitosis, the vegetative cell continues to grow and accumulate the reserve food in the form of the starch and fat oils also. It becomes rich in the protein and its nucleus may undergoes the endoduplication, become highly polyploid. So vacuole disappear, the nuclear membrane became highly convoluted. At maturity stage, the nucleus often lacks a nucleolus. So the generative cell. So cytoplasm of generative cell contains usually the cell organelle like mitochondria, ribosomes, endoplasmic reticulum, some microtubules and dictioles also there, dictyosomes. So plastids are absent in the some species, some are the present. So it is elongates to such as extent that is appear were formed at maturity. The elongated shape probably facilitate its movement to the pollen tube. So division of the that is the generative cell. See here, the generative cell undergoes to the mitotic division to form the two sperms. So this may occur when the pollen grain at the uh, in the anther. So thus pollen grain may be three cell or two cell at the anthesis. In most of the angiosperms, pollen are in the two-celled stage only. So some families means Asteraceae, Brassicaceae, Poaceae, they are three-celled stage also we can see. Okay. So here in the two-celled pollen grain is division of generative cell occur after the uh, alighted embryo sac. Still, in the another case, the postpone to the time when the pollen tube has reached to the embryo sac. So, in some generative cell are more or less prolonged mitosis during the period. The generative cell undergoes to the elongation to acquire its characteristic vermiform appearance are there. Wow. So, in some species with binucleate pollen grain, the division of generative cell is postponed until the pollen germination. It passes through the stage of the temporary dormancy. So here we can see the some male dimorphism is also there. So male germ unit was first discovered by the Thomas et al. In 1984, which is generalized the feature of male germ unit, that is the connected sperm is close associated with the vegetative nuclei. So this one is observed in the Plumbago Zeylanica and Brassica species. They are the here dimorphic condition is appear. So one sperm is large and containing almost all the mitochondria, while the small sperm contain near the plastids. So some species also isomorphic is also there. A common cross wall aggregation of plasma membrane links the two sperms, and larger sperm is also connected with the vegetative nucleus by cytoplasmic extension. Thus, a three-celled male germ unit is formed. Once the male germ unit is recognized, it travels through the pollen tube as one entity. On reaching the volume, the vegetative nuclei and sperm get separated. The density of each sperm is also predominated. Okay. So here, no need of the gene expression during the male gametophyte development. That is so male sterility. Coming to the male sterility. So it is the absence or lack of the fertility pole in male plants. Such male sterility plants plays an important role in the plant breeding techniques, because the male sterility plants can be used in the female parent with the emasculation and facilities easy to cross pollination without bagging. Means in the plant breeding technique, here we can use these male sterile plants. Means when we use in the cross pollination, That time is very used. So here some answer is the suppressions, abortion and polyploid, pistoploid if the way are to is. So aberrant meiosis or gametophytic development sometimes determine the gametophyte, determine the sporophyte. So sometimes anther dehiscence failure. So these are the some fungal or viral infections. So these cause the male sterility. Okay. So here in the coming to the pollen germination or pollen tube growth, here you can see. So sticking and hydration means pollen grains are transported to the stigma by various agents are there, like wind, insects, or water, animals. Mainly the wind and some insects means honeybees, especially in honeybees, there are involved in the pollen transportation. So honeybees are very good pollinators, which are very useful in the cross pollination. So and some extent this depends on the pollen size, mass, and sculpture. So the first interaction between the pollen and stigma is sticking. So the pollen is settled on the stigma. So which is highly viscosity and thickness of the stigma. The factors involve surface of tension, wind force, electrostatic force, and gravity. So these are there. So pollen grain swell in the presence of water, indicating the rapid uptake. So water passes through the exine and it take up by the intine and cell. The full hydration of pollen is not necessarily prerequired for the germination. So pollen tube formation and growth here. So after sticking, the pollen germination starts with the imbibition. So the pollen is fully developed. Germination starts quickly. For pollen germination, water and highly humidity is necessary. In some cases, nutrients, mostly carbohydrates, minerals, and also present. Some are the polysiphonous pollen grains also means more than one tube are germinated. So up to 10 pollen tubes have been developed. Some Althea rosea and Malva around 14 pollen tubes are germinated from the single pollen. So all the content of the pollen grains move into the pollen tube. So active growth, the pollen tube generally restricted to the tip region. So most of the cytoplasm form at the grain is in the tip region and large vesicule fill the grain. So callus plug is also formed in the basal region of the tube and they are restrict the backward movement of the cytoplasm from the tip. So because of the formation of the callus plug, the pollen tube of mature grain is subdivided into many compartments. So callus plug starts originating from the wall of the tube, grows towards the center and reduce the lumen and finally seals the tube. Small amount of the protoplasm left in the grain gradually disappears. So coming to the Here. So, pollen tube wall, which is consist of cellulose and pectin also there. Some specific tobacco plant, the pollen tube consists of two layers are there. So that outer primary wall is the pectocellulic and inner secondary wall consists of pectin, cellulose and callus is also there. So here some the factors affecting on the pollen germination and formation of the pollen tube also. What are the here some effects also there? What are the here in vitro studies we can see here relative humidity and carbohydrates, boron effect, these are there. So highly relative humidity is very essential for the pollen germination. It has been observed in the germination of pollen occur quickly in the standard atmosphere. So come to the carbohydrates. The source of sugar is also essential for the germination of the pollen in the nutrient medium. Carbohydrates are also more essential. Sucrose is found in the best sugar for the prompt of the pollen germination and pollen tube growth. So coming to the boron and calcium enzymes, hormones, this also boron that is the essential for the help in the pollen germination and growth of the pollen tube. So in the mature pollen are deficiently of the boron, hence is the additional enhance the pollen germination. So boron deficiency is compensated by the stigma and style. The presence of boron, the medium preserve the bursting the pollen and also helps in the biosynthesis of carbohydrates. So another in calcium is also calcium element also their enzymes. These are the very essential for the pollen tube. So coming to the here we can see the pathway. So here pollen tube pathway of the pollen tube enter into the embryo sac. Here after the pollen germination formation of the pollen tube for the growth of the pollen tube. So continuously slim passage is required. So this is the mucilage originated below the cuticle outer wall of the epidermis. So that is the end time. The time is required between the pollination and fertilization depending on the rate of the growth of the pollen tube and distance it is traveled. So there are the here different types of pollen tube reach the ovary based on their mode of the entry of the pollen tube into the ovule. There are here three types of the fertilization process are there. There is the porogamy, chalajogamy, and mesogamy. Porogamy means that is the most of the pollen tube enter into the ovule through the micropylar region. From that micropylar region, the pollen tube enter into the ovary. So means that is ovule. So next one is the chalajogamy. Means from the chalajal region, the pollen tube enter into the chalajal region. We can see in the Casuarina. So that character we can see in the Casuarina family, Casuarinaceae. So that is the uh, from the chalajal region. Third one is the mesogamy. In this type, mesogamy means the pollen tube enter into the ovule through the funicles, through the funicle across the integments in the pollen tube to the ovule. This we can see the mesogamy character. Okay. So coming to the here pollen tube enter into the embryo sac. So see here that is the pollen tube enter into the embryo sac. We can see here. So after the passage of filiform apertures of the pollen tube grows into one of these synergies. So these penetrate synergies shows the sign of the degeneration of before the entry of the pollen tube, but after pollination, they are increasing the concentration of gibberellic acid is supported to the possible cause of the breakdown of the synergies in the plant like cotton. So discharges occur through the pore on the pollen tube, which may be terminal or subterminal in the position. So the discharges occur in the second in some plants. The pollen tube is forked and one branch in the directed towards the egg and other towards the secondary nucleus. So here pollen tube discharges and sperms. So another one is the pollen storage. This is a very important some nowadays. Pollen storage is important. So that is the effect of pollination occur throughout the various agents such as wind, water, animals, birds, insects also there. So this one is the depending on the life of the pollen is very short in some grasses. Example, passpal, the loss is capacity germinate within 30 minutes. Okay. Some are the 24 hours that is there. So earliest references the handling the storage of the pollen wall at the date palm. Male inflorescence date palm were considered the most important commerce in the uh, Middle East around 400 years ago. So dry and cold storage. That one is the longevity of the pollen is covered the several factors such as the time, temperature, relative humidity, and light. So this one is also very some effect in the pollen preservation, cryo storage or cryo preservation. We can use the liquid nitrogen around minus 180 degrees for the long period in the presence of liquid nitrogen, which is stored around the fertility here 360 days they have. Okay. Uh, Here pollen allergy is also present. So here pollen allergy. This is the small topic. Pollen and spore allergy. This is also there. Some fungal spores and pollen grains initiate the allergic response to the susceptible individuals. So allergy people that is the react to the potential allergens being the hypersensitivity to then cause the several type of eye and skin and respiratory disorders. Skin allergies also there. These are some itch erupting of eyes. There are two types of major common respiratory allergies are there. That is asthma and hay fever. So asthma that is the respiratory disorders. Hay fever also. So allergens in the pollen grains contain protein and glycoproteins are there. Respiratory allergens is required acquire the hypersensitivity reaction to specific components of the air aerospora. So these allergens of pollen grains are located in the extracellular wall side and intine and exine. This can be established using the fluorescent antibody methods. Okay. So another that is the pollen embryos. This one is some pollen embryos also there. Hmm. So pollen embryos, this is the short topic. The pollen embryos are developmentally similar to the zygotic embryos under the appropriate cultural condition. They can mature and accumulate seed specific storage products in the accompanied manner. Such embryos develop directly to the plants, although plants may arise from secondary structure. Double haploid plants are produced through the chromosome doubling techniques. These homozygous plants are useful in the plant breeding and genetic studies. In addition, the haploid embryos are used in the mutant isolation, gene transfer, studies of storage, product biochemistry, and physiological aspects of embryo maturation. So here the development of polyembryo affected the various physiological condition and culture in the media, divide of the hormones. So that is the here we can see the datura, which is the failure to form the embryos is regardless of whether they are culture and liquid sodium medium. The pollen isolated from anther and preculture and solid medium did form the embryos. So this one also there. So anther culture is the technique for the culturing anther. So here aseptic in vitro conditions raise the haploid plantlets. So here also these applied plants also they are in the used in the plant breeding technique. So then the plant breeders initiated haploid plants because they're either spontaneous doubling or the application of chemical colchin or double chromosome number gives us the homozygous diploid plants. In this technique, the immature pollen is made to divide and grow into callus or embryonic tissues to produce the haploid either through the organogenesis or embryogenesis. This is also there, right? So coming to the female gametophyte here. So megasporangium is nothing but ovule. So it is the protective coat and namely integments that is known as ovule. So ovule is the forerunner of the seed. So therefore, the study of megasporangium or ovule is the most important is understanding the structure function of the seed. So here that is the ovule is consists of nucellus enveloped by one or two integments are there. That is we call after seed coat is formed. So which is found attached to the placenta on the inner wall of the ovary or by the stalk known as the funicle. The part of the funicle that runs parallel with the nucellus is called as a raphe. An ovule ready for the fertilization consists of nucellus enveloped by the one or two integments. So leaving one small opening place is apicalium, that is the micropylar region, and which is passage for the entry of the pollen tube. So when the chalagel, that opposition of the micropylar region, which is the integument is inserted, and funicle, raphe, chalagel, nucellus form the continuous tissues on the central axis of the ovule, and no sharp demarcation drawn between them. So here types of ovules also discuss here different types of the based means the position of micropylar and funicle and chalagel. So position of the micropylar, funicle and chalazal position. So based on these positions here in the angiosperms, 6 types of the ovule types are present. The first one is the arthropous ovule. So arthropous means they are directly means here micropylar, funicle and chalazal region. They are the straight lines are there. So here in the micropylar region, funicle and chalazal, they are least the straight line that ovules we call as the arthropous ovules. And another one is the anatropous ovules. Embryo sac, this is the embryo structure is there. And another one is the anatropous ovules, means the ovule became the completely inverted and the result which is the micropylar and funicle lies very close to each other. That one we call as the anatropous ovules. And hemianatropous. In this, funicle lies at right angle to the nucellus and integments. Right angle to the right angle to the nucellus and integments. And campylotropous ovule. The ovule is curved in shape. Okay. So in the amphitropous ovule, the curvature of ovule is more. They are pronounced here. The curvature also extend to the embryo sac, which is became the embryo sac, which became a horseshoe shaped amphitropous. Sarcinotrophus ovule. So this is the peculiar type of ovule, and here what happened here, that is the funicle to formed another integument, means the complete circle around the ovule, but the free from the except of the small area of attachment at the end of the funicle. That one we call as a sarcinate type of the ovules are there. So few minutes, wait. Thank you. So here, so coming to the integments, different 2 or 3 integments are which is covering the protective layer. In angiosperms, there are the ovule consist of 1 or 2 or sometimes no integments also we can see. So on the basin of there are the termed as a unitegment, bitegmentic, and ategmatic ovals are present. Unitegmic, bitegmic, or either ategmic means no integments are there. So these are there. So coming to the endothelium, this one is the endothelium means gametopetale means gametopetale family members, that is unitegmic or tenunucleate ovules are seen. These integument enclosed rapidly. Acquire dense cytoplasm shows the prominent nuclei and differentiation into endothelium. So otherwise known as a integumentary tapetum. So this is also you can see. So embryo sac is nothing but male gametophyte here. You can see that in the embryo sac here there are the abutator. So obturator, this one is also the association with the function of direct to the pollen tube toward the micropylar. This one is called as a abutator. Uh, so obturator, this obturator shows the great variation in the morphology, origin, and anatomy of the development. In the ovals with the distinct funicule, obturator is generally funicular in origin. In sessile ovals, that is the combination of placenta and funicule. This is the obturator develops on one side of the funicule. So obturator is partially funicular and partially placental region. The cells of the obturator are dense cytoplasmic and rich in endoplasmic reticulum, dictyosomes, vesicles, and other organelles. These cells are produce the surface extricate and provide the nutrition, mechanical and chemical guidance. Their pollen tube, the obturator is divided of any vascular tissues and cells shrink and disappear out after fertilization. So nucellus, which is the apex of the nucellar ovular primordium. So this the majority of the separation means nucellus is present the single layer of the cells, namely nucellar epidermis, and enclose the megaspore mother cells. Such ovals where the megaspore cells is hypodermal in region and in the nucellar epidermis around it remains a single layer are termed as a 10 uninucleate. So coming to the megasporogenesis. So this is the megasporogenesis. What here? Uh, the archisporium is the hypodermal in region. A cell of the nucellus situated below the epidermis became the more conspicuous by its larger size, dense cytoplasm, and prominent nucleus and functions of archisporium. So archisporial cells may divide to form the primary perichial and primary archisporial cells. It may directly function as a megaspore mother cell. The primary perichial cell may remain the undivided or undergo further divisions and form a very variable number of wall layers. So primary sporogenous cells generally function as a megaspore mother cell without any further divisions. So after megaspore mother cell undergoes to the meiotic division, form the four tetrads. The first division is always transverse and results in the formation of dyad. The second division is also transverse and result in the formation of linear tetrad four megaspores. So sometimes vertical division is in the micropylar dyad and transverse division in the chalagel dyad. So there are formed in the T-shaped manner. So here there are the types of embryo sacs. So types of embryo sacs we can discuss here. So there are the depending on the number of megaspore the development of embryo sac. Three main groups embryo sac develop can be here we can see that is the monosporic, bisporic and tetrasporic type. So here first only one fourth megaspore takes part in the development of megametaphyte. In the second two megaspores nucleus part in the formation of this megaspore development and the third one is the tetra means four of megaspores are involved in the formation of the megasporogenesis. So one by one we have to discuss here monosporic type. So the nuclei of this type of embryo sac is generally alike as the derived by the mitosis of a single nuclei. There are again here monosporic, again here polygonal type and inothera type of embryo sacs are there. So polygonal type firstly discussed by the Strasburger in 1879. So polygonum that embryo sac eight nuclei are present. See here polygonum type of embryo sac. Monosporic embryos, bisporic and tetrasporic. So here that is the polygonal type. Here eight nuclei are present. And inothera type. Here inothera type matures sac in the four nuclei. So the inothera lamarckiana, this type of embryo sac is usually formed the micropylar megaspore of the tetrad, which undergoes to the only two nuclear divisions. So instead of the usual three occurrence of polygonum type, the organization of the mature embryo sac is an egg aperture and an uninucleated central cell. Since there is no third division and all the nucleus are situated in the micropylar part of the developing embryo sac, there is neither lower polar nucleus or any antipodal cells. This type of embryo sac development is found to be characteristic and a constant feature of the entire family Onagraceae. So coming to the bisporic type of embryo sac. So in the bisporic, in this type embryo sac develop the first meiotic division is accompanied by the wall formation. So the dyad is formed. Only one of dyad undergoes to the second meiotic division and other degenerated. So the functional dyad cell wall formation does not occur uh, after meiosis two, and both the resulting the megaspore nuclei contribute to formation of the embryo sac. So each megaspore nuclei undergoes two mitotic divisions forming the 8 nucleate embryos are finally organization is similar to the polygonum type. So coming to the allium type here. So there are the different types of allium type and endemion type. Bisporic embryo sac. Here two categories are there: alien type and endemion type. So in this alien type here, this alien type was first of all discussed by the Strasburger. The megaspore mother cell undergoes the mitodivision one, resulting the dyads are formed. So of these two dyads, the chalazal became function while the micropyle degenerated. The nucleus of the functional dyad cells became the undergoes the mitodivision to form, forming the two nucleus. These two nucleus move to the opposite pole on the long axis, are separated by the vacuole. So these two nuclei divide to form the four, and then the eight nucleus is formed as the polar nuclei. So in this type of embryo sac, you can see in the so means Butamiaceae and Alismidaceae Viscaceae family members. So endemion type. So in this type was first described the endemion Hispanicus. In this type, what here spore mother cell undergoes the meiosis one formed the dyad. Micropylar dyad is functional while chalazal dyad cell undergoes to the degenerates. So that micropylar nucleus divide to form the 2 to 4 and finally 8 nucleate embryo sac comprising egg to synergies to polar regions and 3 are the antipodals. That is the endemion type of bisporic type. So coming to the tetrasporic type of embryo cell. The tetrasporic type of embryo sac here, in this group, meiotic divisions are not accompanied by the cell plate formation. Hence, all the four haploid nucleus at the end of the meiosis enclosed in the common cytoplasm, forming the cenomegaspore. So all the four nucleus are formed the that is the cenomegaspore take part in the formation of the embryo sac. Hence, the tetrasporic embryo sac is more heterogeneous than the bisporic embryo sac. So, after these tetrasporic embryo sac, again divided into different categories are there: Adaxia, Peperomia, Pinia, Rusa, Fritillaria, Plumbagella, and Plumbago. Based on these characters, means Maheshwari is these tetrasporic embryo sac divided into further categories are there. So what is Adaxia? Adaxa type of tetrasporic embryo sac. So Adaxa type of tetrasporic embryo sac, which is the first discovered by the Johnson in Adaxa uh, Muchetellina, and which is uh, there are the five only the five genera which is developed the so in this type what here there is a nucleus of megaspore take to 2 plus 2 arrangements. All the nuclei of a megaspore undergoes the one or more division to form the 8 nucleate. So the mature female gametophyte has a normal egg aperture. There are the antipodal and two polar nucleus is present. Another type of the tetrasporic peperomia. Adaxa, Peperomia, Pinia, Drusa, Fertilaria, Plumbagella, and Plumbago. So, Peperomia. So, Peperomia is reported by the Campbell and Johnson in Peperomia pellucida. Here, what? Here, there are the tetraspore adherents are resulting the 16 nucleus are microspore formed. So, according to Johnson, the finally organization of the micropilar end from the egg and synergies 8 fuse to form the secondary nucleus and the remaining 6 are cut off the periphery of the embryo sac. Pinia type. So, pinia type of the tetrasporic embryo sac. There are the Pinaceae family. Here, megaspore takes in the tetrapolar arrangements and divide to twice to form the 16 nucleated embryo sac. 16 nucleated embryo sacs are there. So, see here, Pinaceae type, Pinia. So, the 16 nucleus are arranged crosswise, 3 nuclei of each. Uh, there are the 4 triads and polar nuclei. Generally, the central cell of the micropilar triad along functions as eggs. The central cell of the other triads also simulate the eggs. This type of embryo sac developed in the found characteristic of the family Pinaceae, and here some of the Malpighiaceae, Euphorbiaceae family. And another type that is the Plumbago and Drusa, Fertilaria. These are there. Plumbagella. These are the some types of okay. So tetrasporic embryo sacs. So here we can see the here embryo sac structure of the ultra structure of the embryo sac. So now ultra structure of the embryo sac here first of all described by the female gametophyte in angiosperms by the Strasburger. So Strasburger is the first to describe the female gametophyte. Okay. So here what? Here angiosperm polygonal type, allium type, endymion, odoxan, Fertilaria type. These are the angiosperm type. Here the embryo sac consists of 7 cells and egg cells, 2 synergies, the central cells. These 3 antipodal cells are there. So variation of the number of might have result from the absence of one or both synergies, reduced and increased in the number of antipodal cells. Generally, the nuclear embryo sac except the central cell are haploid. The central cell consists of 2 haploid nuclei called as the polar nuclei, which latter form the diploid secondary nucleus. So synergies also there. 2 synergies are there. These are micropilar and micropilar end and embryo sac. So 2 synergies if the present attached one another and also cover the egg. So these synergies is the structure one synergies degradation period to arrival of the pollen tube. So egg apertures also there. Egg cell occupy the central position in the egg aperture. So here in this is surrounded by the synergis and central cell. The egg cell also has a partial cell wall. The egg cell wall is thickest at the micropellar region and gradually thins toward the chalazal region. So chalazal half is limited by the plasma membrane only. The lateral wall of the egg cell joined the central cell at the majority at the micropellar end and so actually at the end of the central. So antipodals also there. This is the antipodals. So this is the structure. This is the mature embryo sac. Chalazal pole. So this is the chalazal end and integments, right? So antipodal astor is also formed some. So this is the types of already we have to discuss the types of ovals, arthrotrophus, anatrophus, semi-anatrophus, comphylotrophus, and saxonotrophus ovals. That is we already discussed that one, right? So this is the first class we have to discuss transfer section of anther. Anther lobes are there. This is the lobes means epidermis, endothelium, middle layer, and tapetum walls are there. This is the anther here transfer section of the anther. Okay. So, thank you everybody. Thank you to attend the classes, right?
Thank you,
So
sir.
thank you. So remaining class we have to continue next. Okay. Thank you.